Thank you so much for having me here. My research interest is looking into the influence of novelty and prior knowledge in memory networks in rats and in humans. So today I'm going to share some of my basic neuroscience studies, in prior knowledge and also in novelty, and how does that help us for information seeking and also in memory. So this may be a slightly different perspective than maybe what you used to. So yeah, hope you enjoy. So what my research interest is in what makes memory last. So how is it that we have some information remember well, but not the others? So in my lab, we use different approaches from and using different species too, from rodents to humans. And there are different factors that affect our information seeking also with memory. So sometimes maybe with emotions, sometimes with attention, right? And sometimes you may find new information which is more easily to remember when you can fit into what you already know. So today we're going to focus on two main factors. The first one is novelty, like an element of surprise, and the other is prior knowledge. So I thought maybe a relevant example for psychologists or neuroscientists is perhaps attending a talk, for example, like this one, although we're not in the Tom Riley building. But let's just say for those of you who are attending this talk, you may forget what I'm saying probably by the end of tomorrow, let's just say. However, if someone dressed up as the Beatles, coming in, maybe handed you some nice Earl Grey tea or mince pies or biscuits, you may remember this moment better because it's novel, like a surprise element for you all. And consequently, you may remember what I'm talking about, like novelty and memory. Another daily example for us would be where did I park my car, right? So you go to a shopping mall, maybe you park your car at a shopping mall, and after a few hours later, you got to find your car. So in a similar vein, I use an animal model, which is this, we call this arena, where the animals have to find food. So they have to remember where is the location, and they have to find food here. So in this arena, you can see that there are 4 starting locations. So it's north, east, south, and west. So you can imagine this is more maybe like a shopping mall, right? You can you have different entrances. On the surface of the arena, there are a few holes here where I will insert the sand wells. So this is the look of the sand well. So I buried food pellets in there and covered it with sand. So these sand wells are inserted into the arena. So the animals have to find the correct location and when they can find the food. Also, you can see that there are different landmarks, so there are different intra-arena and also extra-arena cues. So this is to help the animals, to find their way. Versus in wayfinding or spatial navigation is important, to have landmarks. So this setup allowed us to look into the spatial navigation paradigm and then looking into the everyday memory. So in the encoding trial, I put the sand wells and they buried the correct location, the correct pellets into correct locations. And in the encoding trial, the animals have to explore what's going on. So they have to find the pellets and they have to collect the pellets. And after a time delay, usually 30 minutes, and then I will perform I'll give them a choice trial. So in the choice trial, they have to find where is the correct location. So only the correct location will be rewarded. So the other wells are they have sand in it, but they are not rewarded. So on the surface, the animals couldn't know where is the correct location. They actually have to dig it. So they need to remember the location. So the next day, the location will change. So it will change to a different location. So you can imagine that maybe when you park your car, right? So you may not be able to always park at the same spots every day. So what I would like to show you is a video of the animals finding the correct location, finding the correct pellets. So in this clip, the animals already experienced the encoding trial on that day. So he's starting from one of the starting locations, right? Hesitate, thinking about where should I go, and then go into the arena today. So he thought that this location is the correct location. So he's digging. It's quite deep, like the pallet's buried quite deep, so it takes a bit of time. And he knows that this is not the correct locations. This is the correct location. And then he took the pallet and then eat at the start box. Okay. And you can see that there are some intramaze and also extramaze arena cues. This is to help them to navigate the place. The reason the animals, going to a different stat box to eat the pellets because we would like them, to explore the arena more and using the spatial cues. So it's not just doing this by egocentric, not just like turning left and turning right. It's actually pay attention, to the cues to explore the arena. So here is showing the protocol. So this is the encoding trial. So with one group of animals, so after 30 minutes, we did a probe test, and the other one is after 24 hours. So what is the probe test? So in the probe test, there are no reward pellets, but we are testing whether animals will dig in the correct location versus the incorrect one. So here's the data. This is plotting the dig time versus like the delay. So with the 30 minutes group, you can see that the animals dig preferentially at the correct location, while the 24 hours group, the memory is at chance level. So this gives us the paradigm to look into, right? So they have really good short-term memory, right? But after like 24 hours, they forgot. So here we're thinking of, okay, right, so this is paradigm to look into this short-term memory. Could we, find any ways, to boost this like weak memory here? So it's supposed to be like in 24 hours they forget. So could we use like different methods, to boost this memory? So what we did was after the encoding trials, we let the animals to explore like a novelty arena. So what happens is in this arena, there will be different other substrates so that animals have an experience. So it's just 5 minutes exploration, and then we test them. So here what we see is that with novelty, the animals remember what's going on with the correct location, and without the novelty, like they couldn't. So this is an example I made is like, imagine there was some surprise element. So you may remember, this event and consequently, events happening around this time. So when we look into the literature, looking into more the basic neuroscience paradigm, so what we found is that there are different brain regions that involved in detecting novelty. So hippocampus is one, and also there are different regions looking into like locus coeruleus and also ventral-tegmental area. So a lot of literature is looking into with the ventral-tegmental area is that it's thought to be exclusively source of the hippocampal dopamine. However, it's only recently, like with locus coeruleus, like is the actual one of the key area brain regions, involved in the dopamine release, which it boosts like memory retention. So here is a sagittal section of a rat brain. So here is the anterior part and here is the posterior part. So what is illustrating here is that both region, used to be believed that with literature used to believe that both regions are involved. And now actually when we look into the recent literature, you can see that like LC has more projections, to hippocampus than the VTA. Right. So in order to further look into this, what we use a tool we call like Optogenetics to see if we activate this locus coeruleus, could we mimic the effect of the novelty enhancement? Okay. So if you remember that, so like with the 24-hour memory, we have like a weak memory, and by using novelty surprise elements, we can actually enhance this memory, right? So now we are thinking, could we replace this like novelty enhancement by using like optogenetic stimulation in the locus coeruleus? So what we did was introduce like light sensitive opsin crimson like into the locus coeruleus. So optogenetics is a tool which is allowed us like to shine lights into specific neurons, where you can actually activate or inactivate like certain like neurons in specific brain areas. So here is again, this is sagittal like section of the animal brain, with this is the anterior and this is the posterior part. So we put like optrode into the LC, which is include like optic fiber and electrodes, and we record some of the light activity in LC. Also, we implanted some cannula. So this is a drug cannula in hippocampus where we can actually inject like drugs into the hippocampus, and also optic cannula into the LC area. Oops. And here you can see that this is an LC expressions where we're overlapping with the TH neurons and also neurons expressing like channel rhodopsin. So basically looking into what's going on in the LC area. And here is showing the histology. So this part highlighting here is the LC cannula. This is a coronal section of what's going on in the rat's brain. And here is the hippocampus cannula, like a histology, okay? So this basically set the scene for us to look into the optogenetics happening in the LC. Here I'm showing you like a video of what's going on. So what is the stimulation? So this is basically mimic what happened in the novelty experience. Like so you can see that the animal is very, very calm, and we have like some stimulation going on. And we've been habituated animals, so this is like no pain, like, you know, for the animals, it's like experiencing what's going on. So if you remember the protocol that during the encoding trials, the animal learns like where is the correct location, and then afterwards, instead of the novel explorations, so what we did is the optogenetic, so just stimulate with just 5 minutes with the LC area, and then we test the animals again with the probe trout to see whether they remember it or not. So what we found is that with the channel rhodopsin with the lights on, where you see like the optogenetic stimulating in the LC area, the animals remember, you know, the correct location, while the other control groups, they couldn't remember. So with this protocol, what we can look into is that we successfully, you know, stimulate, you know, the LC neurons, which we can mimic the novelty effect. So then next, we're interested to see, you know, whether, you know, if we block, you know, the hippocampus, where it is one of the like the dopamine source area, to see whether we can block this effect, right? So we've used optogenetics to simulate the LC. We managed to see like a boost of the memory. And now if we use some drugs, which is shearing, which is the dopamine D1, D5 receptor antagonist, to see whether we can block this like effect. So what happened with the protocol is that we did the drug infusion in the hippocampus, and afterwards we did optogenetic stimulation, and then the animals like encode what's going on in the correct finding. You remember, you know, the correct locations, and then we test the animals. So we did it like before and after. We don't find any differences between if we do it before or after, but what we found is that if we put shearing here, we managed to block, you know, the effect. So to sum up, what we found is that using this everyday memory protocol, we managed, you know, to look into enhance like the memory of the animals by like using the novelty effect, and also using optogenetic stimulation in LC, we managed to mimic the effect. But if we block it in the hippocampus with the dopamine like receptors, we managed to see that's the deficit in this with this memory protocol. So what I'm trying to say here is that like novelty, and also novelty is one of the important like, you know, factors that affect our everyday memory. So now I would like to talk about another factors, which is prior knowledge. And this may be more familiarized like with you all. So have you all heard about like prior knowledge and schema? Some of you maybe guess, like yeah, so this is a concept that's developed and introduced by a different psychologist for a while. So, for example, Bartlett, Piaget, and Bransford. This is a specific knowledge structure, a cognitive framework that helps like organize and interpret informations. So it has been developed, and there's a lot of like human psychology literatures on it. However, there's not a lot of neuroscientists looking into the neurobiology of schema. So that is how I got really interested to look into neurobiology side of it. So I'm using an example, like you all familiarized with the spatial map in Liverpool, I guess, yeah. So here we are, like at the James Parsons building, right? Like a Tom Riley building, like yes. And I'm noticing the Christmas market, right? It's the St. George Hall, right? So for those of you really familiarized with Liverpool, you know the area, you have a really good spatial map. So you have like a good knowledge here. So now if I tell you, right, they have closed this Christmas market and put it in, to print the stock, right? So for those of you, really familiar with Liverpool, like, spatial map, oh, I get it, like, remember this instantly. But however, if I'm not really familiar, like for myself, like I probably remember the wrong dog, probably remember like the Albert dog. So like a good, prior knowledge is helps, to incorporate the new information easier. So that is the base of like schema, which when new information can incorporate, what you already know, it makes things easier. So how did I investigate, into the neurobiology of memory? Again, I used the event arena. So with the event arena is where event happens in the arena, right? So again, this is what I showed you before. So there are 4 starting locations. You can imagine this is more like a shopping mall again, right? So and these are the halls, like where are maybe different stalls, like different cafes in the shopping mall. You have different like types of food, right? So the animals have to enter from different entrances. So there's 4 starting location. And this time, instead of just one correct location, there are 6 locations. So the animals have to associate with 6 different flavors. Okay, so for example, this is one of the correct location. And here is the spatial arrangement of the schema. So in location 1, there is a lime flavor, okay? And in location 2, like with fruit palettes with banana flavor. And here is grapes. And we even have a pina colada flavor, and the animals love it. So what happens in a trial is that I'll give the animals a food palette, a cue, like in one of the certain location. And then again, the animals will have to explore the correct location. So only the correct location will be rewarded, okay? However, some of you may ask, well, maybe they could smell and do it, but with this sandwell, we actually put, enough palettes. So this is on the top side, which is an accessible like layer that they could actually get the palettes, but also in the inaccessible size part, we also put palettes so that it's all smell the same. So basically with the incorrect well, we've got all the palettes at the bottom, so they can't reach, but it's all the sandwells found the same. So it's to make sure that it's not doing this, by arbitrary use. It's like they actually have to remember where is the correct location. Okay. So it takes the animal, a while to learn. So it takes them about like a month, at least, to learn this. So in between that, we did some probe trials. So the probe trial is one way to show that they actually learn, the test. So in the probe trial, again, there is no rewarded pellets, but we are measuring the dating time, the time that is spent in the correct location versus the incorrect. Okay, so here is the correct location, and here is the incorrect one. So in the beginning, after like just one day, they're at chance level, but by month time, they remember this. So they remember this 6 paired associate location. So the theory of like schema is that, so once they learn, what's going on, right? If we include new information in there, if they can remember really quickly. So it's this rapid memory consolidation. Because with the usual system memory consolidation, it takes months. So we'll see whether this, protocol works. So once they will know that they learn the schema, what we did is we close like two sound wells. Okay, so imagine it's like close to restaurants. Okay, and then I introduced two new ones right here. So it's a different flavor, it's a lemon and cherry, and it's a different location. And this is only with one trial. Okay. So with that, the animals learn that with one trial, but this only happens because they have like the backdrop, like they have the schema. Because if you just introduce like one paired associate to them, like they would not remember that quickly. So the next questions we interested is to look into what happened in the brain, right? So we know that like hippocampus is important, to remember new information. And this is like a hippocampal dependent task. So, however, what's going on? What's happened? So what we did was that we divided the animals into two groups. So once they learned these new paired associates, we divided them into two groups and we performed like hippocampal lesions. So with one group is that 3 hours later, like we did like a hippocampal lesions, and then the other group is like in 40 hours. So you may wonder that why do you want to understand, like you have two groups, right? So the reason for that is because we would like to know whether this information actually transfer from hippocampus to the other cortical area or not. Because with the system center consolidation theory is that initially we required a hippocampus, but by time goes by, this is more from like the cortical area. So we would like to see with this really rapid memory consolidation, does that like where does this go? Does this information goes outside to the hippocampus a lot or not? So what we found is that with the 3 hours group, like with the hippocampal lesion group, they couldn't remember what's going on with the new paired associate, but with the 48 hours groups, they remember. That means the information transfer, outside the hippocampus between 3 to 48 hours. Okay. So then the next question we would like to know is that where does this information goes to? So using immediate early genes, which is a neuronal marker, counted like different brain regions. So what I found is that with the prefrontal cortex, which is important for the executive functions, is heavily involved in this in this rapid memory consolidation. So you can see that here with the new paired associates group, like which with the prefrontal cortex is heavily involved. So following up with more recent studies looking into different brain regions. So these are like more further analysis with different brain regions. For example, dentigerous, CA3, CA1 is all hippocampus. And here, prelimbic and anterior cingulate is more like prefrontal cortex. What we're seeing is that the prefrontal cortex is important for this rapid memory consolidation along with the hippocampus. So you may think that this is like all good, right? Like, so how does that maybe link to also in human case, right? So I've just used another example, like to talk about spatial schema. So for my understanding, LJMU they have like two main campuses, or it's expanding now. Like yeah, it's expanding. Okay, so we've got this like city campus, right, and the other is the Mount Pleasant campus, right, where the yesterday we were delivered to neuroscience like lecture holding, right? So this you probably have like two spatial schemas mapped, right? So you know what's going on with different buildings and different campuses. So in the animal studies, and also did like a consistent schema map and versus an inconsistent. Okay, so a consistent maps, which means the animals have to learn this like flavor map consistently, throughout a month. With the inconsistent one, they have to learn a map also, but within this map, like the flavors location changes. Okay, so this is, although the spatial arrangement is the same, but the actual content it is changes. So this the reason I'm looking into this is to see that is that only like a schema will will help new information, or does it has to be more like a consistent schema, the schema that actually makes sense to you in order to incorporate the new information. So I found that with the animals, if they've trained with a consistent schema, they remember the original paired associate. And then with the new paired associate, so again, it's like close like two locations and then introduce two new ones. They actually remember only with the consistent map versus like, the inconsistent one. Okay, so what I'm developing now is like the translation of a schema paradigm, in animals, to the virtual reality, like task for humans. So all of us have like maybe shopping experience, right? Like like maybe like shopping, or not. Like yeah. So this is just like a demo showing, if you go to a shopping mall, right, you have like this like corridor, and then you go to the different shops that you may like. So, oops. So what I'm trying to do it here is with my students that we're looking into this novel like human schema paradigm. So this is like a pilot study. So still things are still developing. Okay. So there's some we'd like to show you some unpublished data. So here you all familiarize with the animal task, but right now what we are translating is like the human schema task. So here is the layout from, imagine it like from an over top view, right? So you still have like 4 entrances, okay? So you still have like north, east, south, and west. And then there are different shops. So for example, this shop is selling ties, or like this shop is selling like handbag. So in the beginning of the trials, like the participants will be given like a picture, like let's just say like a handbag, right? So the participant needs to go inside, and they were having like a first person view. So they will not have this like over top view. Okay. So they would go into the shop, and then they have to go and find the correct like item. So I'm still measuring like similar things like for the number of errors, the latency, and also the distance, to travel. So you imagine like, it's still like spatial navigation and looking to what's going on. So here is a pilot video that made by Christina. So the participants first look into like they have to make the handbag, and then they have to enter from one of the starting location. So there are some cues and landmarks, so they have to go into different rooms. So this is a place where have pictures of like shoes. So this is not the correct place. So then the participant needs to go to a different room to have a look. So this is still like a learning exploration like phase. So this shop is selling watches. And yeah, so this is not the correct place. So it's making like 2 errors already. So this is some landmarks there to help them to navigate. And here finally, the participant arrived at the correct location, which is selling bags. But what we want is a handbag. And then the participant needs to learn again, like, every like day they have to learn these 48 trials, like to building a schema. So here it shows what's going on for the trajectory. Again, we are looking from the view from more like an overview from the top. So this is the starting location is as north. So what you've seen just now, the participant explore into one room, which is the wrong one, and then go through the corridor, explore another one, which is like the second shop, which is selling the watches, and then finally going into the correct location. So there are different landmarks here, like helping the participants, to guide them, to know where is the correct location. So here I'm showing you like different maps. If you remember the story about the importance of the consistent versus the inconsistent map, right? So for the one group, we introducing with the consistent map, so they have to learn this like map throughout the all the trials. And the inconsistent map is that they have to learn between like map A and map B. Although the spatial arrangements is the same, but what inside the shop is different. For example, with the consistent map 1, they learn that like handbags is always at this shop, at like shop 4. Let's just say if this is 1, 2, 3, 4. And with the map B, it's going to be in here. So the participants like got to remember like both like maps, okay? And the reason I would like to introduce that is to see when they learn the new shops, like new items in different shop location, whether they could remember it or not. So once they learned, you know, in the first day, they learned what happened in the original pair associates. Then in day 2, they have to learn this two, they have to learn this two new location. So I will close like one shops and the other shops here and introduce like 2 new shops with 2 different items. So if you remember with the animal studies, like I close like 2 Sandwells and introduce like 2 new ones. So it's similar to that. And if you think about with us, maybe when you go shopping, so sometimes, you know, the shop changes, right? So you got like new shops open and some of the shops closed. So here is look into whether with a consistent schema, can the participants, you know, learn better? So here show you some like preliminary data. So first of all, with the consistent map, like the participant remember better and they spend more time explore in the correct locations versus the inconsistent one. And then when I introduce the 2 like new shops, buying like 2 new items. So with the consistent schema group, they can perform better versus the inconsistent one. Okay. So this is still the study is still developing. And so what I would like to show you is that to conclude is that like we have managed to develop like the first like human spatial paired association paradigm, and with the consistent schema, it facilitates like the new paired associate learning. So what is the next step, right? Because this is still, you know, in the beginning, and what we're doing is that we're recruiting like healthy aging like 65 like plus years old people, and we're collaborating this with the U3 from Alton and Omskirk. And also collaborating with Reese Davis, like from the Walton Center, we're also going to look into the early onset dementia patients. So they're going to use this paradigm, and we would like to explore, you know, how this prior knowledge, you know, helps with like the new paired associate learning and also with new learning. Just one more thing I would like to talk about is it's really good like working with like the U3 group because we launched a series of events like since last year, and with this Aging Better with an Active Mind project. So we co-create with the U3 group to look into more about healthy aging issues, for example, like dementia and memory, physical activity and active mind, and emotional health. And recently, we also invited Susanna to give a talk about the importance of touch. So it goes with like a series of like healthy aging events and with different like collaborators. So, and one last project I would like to talk about is understanding schema development in children. Okay, so maybe some of you play Minecraft. Yeah, no, like or knowing some people play. So using Minecraft like as a tools, like we developed this spatial navigation like paradigm. You're probably familiarized with this already, but you probably built the schema already, right? So this is the event arena, okay? So again, there are like 4 starting locations. So what the participant needs to do, you know, is to go into the starting location, you know, break, you know, the blocks, the flavor blocks, and then going into the arena to find the correct blocks. So here I'm showing some snapshot for you. So they have to go into one of the starting locations. So this is a blueberry flavor block. And then afterwards, enter to the arena. Then they can find like different cues, right? Like in the arena, and they have to find the correct location. Again, so you can see that here is the correct location. So then they could need to break the blocks. So it's a like it's a direct, more translational like paradigm, you know, from the animal work to here. And what I would like to know more is that how does like children, you know, form like a schema. So what actually, you know, way, you know, different methods do they use? So after, you know, measuring different like of the number of errors and also distance, we also asked them, you know, could they locate where is the cues? Like, could they locate where is the landmarks? And what we found is that again, when we did like probe trials where we see that with the participants, they made more errors in the beginning, and then by the end of it, they like actually learning the task and actually making like less errors. So what we are going to launch this study is that we're looking into different like consistent schema and inconsistent schema.